this session, we are discussing glandular system in insect. See how a gland develops. Glands develop from a ordinary body cells during the embryonic development. Glands they develop from ordinary body cells during the embryonic development. Certain cells become modified to produce secretion. That cells increase in their size and develop into a specialized glandular tissues are known as glands. See the glandular system in insects. The glandular system in insect is also known as secretory system. It is divided into two major groups based on the presence or absence of the ducts. See that major groups are A. Exocrine glands, that is gland with ducts. Second one is endocrine glands. These are the glands without ducts, ductless glands. Exocrine glands, these glands release their secretions through the ducts onto the body surfaces or into body cavities. Onto body surface mean that wax, pheromones and defensive secretions are secreted on the body surfaces. Into body cavity mean that saliva is released into the mouth cavity. In that way, the exocrine gland have ducts and they release their secretion onto the surface of the insect body or into the body cavities. Next, endocrine glands. These glands are without ducts and release hormone directly into the hemolymph. And hemolymph carries this hormone throughout the insect body. See here, list of exocrine glands, the glands with ducts, and endocrine glands, the glands without duct. Mandibular gland, salivary gland, maxillary gland, pharyngeal glands, frontal glands, setal glands, tenant hair, molting glands, stink glands, asmatoria, androconia, pheromone glands, wax glands, sting gland, lac gland, milk glands. All these are the exocrine glands. They possess duct. Endocrine glands, neurosecretory cells of the brain, corpora cardiaca, corpora allata, prothoracic gland, and Weissman ring. All these are endocrine glands. They do not have ducts in them. They release their secretion directly into the blood. That is hemolymph of the insect. That hemolymph carries their secretion throughout the insect body. Let's see in detail about mandibular glands. See in this image, this is the mandibular gland. Mandibular glands are glands that are associated with the mandibles, that is primary jaws. They develop from the tissues which are nearer to the mandibles during the embryonic development. See their function. Produce secretion near the mouth parts that helps in feeding and lubrication of the food. In queen honeybees, The mandibular gland secretes or produce queen substance, that is pheromone, that control the entire colony activity. Let's go to the next gland, that is salivary gland. See here in this image, this is a salivary gland. Salivary glands are modified labial glands because they're developing from the labium region. That's why they are the modified labial glands. Salivary glands are the modified labial glands because they are developing from the labium region. During the embryonic development, cells from the labium grow inward and form glands. That is nothing but salivary gland. And their duct open below the hypopharynx, that is the tongue. See the function of salivary gland. They secrete saliva, help in digestion, lubricate the food, and aids in swallowing. In caterpillar, like silkworm, the salivary glands are modified into the silk gland. So they mainly produces silk instead of saliva. In that case, the mandibular gland helps in secretion of the saliva for the lubrication of the food particles. When salivary glands are modified into the silk gland, in that case, mandibular glands are helping in secretion of the saliva for the lubrication of the food. The next gland, that is maxillary gland. Maxillary glands are associated with the maxillae, that is accessory mouth parts, secondary jaws. This maxillary gland secrete lubricating substance near the mouth part. See the function of maxillary glands and their secretion. The secretion lubricate mouth parts during the feeding, help in smooth handling and movement of the food, assist in feeding activity by moistening food particles. Next, see in the image, pharyngeal gland found in nurse bees. This pharyngeal gland produces royal jelly, that is bee milk, for larvae and queen development. So pharyngeal glands are there mainly in worker honeybees. Next, frontal gland. See here in France region of the head. Frontal glands are large defensive glands present in the head region of the nosty termites. These glands open through a small pores or nozzle on the front of the head region, and they secrete sticky glue-like defensive fluid that protect the termite colony from the predators such as ants. This sticky secretion traps, repel or irritate enemies. In that way, this nasty termite get protection from their natural enemies. See next, cetal gland. Cetal glands, inside the gland is there, over that seta is there. Cetal glands are associated with hair-like structure, seta or branched projection, that is coli. Inside of this branched structure, a gland is there, that is the cetal gland. Cetal glands are associated with the hair-like structure, seta or branched projections, coli. These glandular hairs contain poisonous or irritant secretions. Produces irritant fluid for defense. Protect the insect from predators by causing irritation or itching on contact. So cecal glands are there in hairy caterpillar and slug caterpillar. Next gland, tenant hair. Tenant hairs are present on the pulvilli of the insect leg. Pulvilli, it is a pad-like structure there at claw region in insect, and this hair secrete a sticky fluid. Tenant hair secrete a sticky fluid, help insect to attach to the smooth surfaces, enable walking on the walls, glass and ceilings. So tenant hairs are there on the pulvilli of housefly. Next, molting gland. Molting gland are the modified epidermal glands or modified epidermal cells. Molting glands are the modified epidermal cells present below the cuticle. They secrete molting fluid during the process of molting or ecdysis. See the function of molting gland secretion that digest and loosen the old cuticle, help the insect shed the old exoskeleton and assist in the formation of the new cuticle. And it is essential for growth because insect can grow only by the process of molting. Therefore, molting is an exocrine gland, that is a modified epidermal cell. Next, stink gland or repugnatorial gland. See here, these are the stink gland in stink bug. Stink gland are defensive glands that produce foul-smelling chemicals. The secretions are released when the insect is disturbed or attacked. That secretion protects the insect from the predators, repel the enemies by bad smell and irritation, and the stink glands are there in stink bug, bed bug, brown marmorated stink bug. Next exocrine gland is asthmateria or forked gland. See, this is the asthmateria. Asthmaterium is an aversible forked defensive gland present in the thoracic region just behind the head of certain caterpillars. It comes out when the larva is disturbed. These glands emit an unpleasant smell or defensive chemical that protect the larvae from predators and natural enemies that repels attacker by foul smell. We can see asthmateria in citrus butterfly larvae and in lime butterfly larvae. Next, androconia or scent scale. See here, patch of scent scale. See here, patch of scent scale. Androconia are specialized scent producing scales present mainly in the male butterflies. The scales release characteristic scent or pheromones. that attract female during mating, help in courtship behavior and species recognition. So these are there in period butterfly and in so many other butterflies. Also Androconia is there. Next, pheromone gland. See here, female moth is releasing pheromone that is detected by male moth. Pheromone glands are the gland present near the abdominal terminalia of many insects. At the abdominal terminalia, in so many insect, pheromone glands are there. They produce and release pheromone, that is the chemical signals. We communicate by means of language, but insect communicate by means of producing chemical signals, that is pheromones. Their secretion attracts mate, help in communication between insects of the same species, and assist in mating behavior. Pheromone glands are seen in female moths, many beetles and butterflies. Wax glands. See here in this image, honeybees have 8 wax glands that produces bee wax. Liquid wax oozes out from these glands and harden once exposed to the air. This liquid becomes hardened when it is exposed to the air. So wax glands are the specialized glands that secrete waxy substances. The wax is released onto the body surfaces through the small pores. That waxy secretion protects the body from water loss, form covering, nest or protective layers. In honeybees, wax is used for comb construction. These hexagonal cells are made by using this wax only. So wax glands are generally seen in honeybees as well as in mealybugs. Next, sting gland. Sting glands are the modified accessory glands. See here, these are the sting glands. These are the modified accessory glands associated with the sting apparatus. They produce and store venom. The secretion defends against enemy, protect the colony, immobilize or injure the attackers. So sting glands are there in worker bees, wasp and western honeybees. Lac gland. Lac glands are the specialized dermal glands present just below the cuticle in case of lac insect. This gland secretes a resinous substance, that is gummy substance known as lac, through the tiny pores onto their body surface. This is the lac. The secretions form a protective covering around the insect, produce commercial lac which is used in varnish, polish and sealing materials. Lac glands are generally there in lac insects. Milk gland. Milk gland are also modified accessory glands present in some viviparous insects. These glands produce nutritive secretions, that is milk, to nourish their developing larvae inside their uterus. Milk glands are generally found in viviparous insect and they produce milk for their developing larvae inside their uterus. This milk gland is generally there in cephate, provide nourishment to larvae during their internal development and support larval growth until their birth. So we have completed detailed discussion about exocrine glands, that is the gland with duct, that is mandibular glands, salivary glands, maxillary glands, pharyngeal glands, frontal glands, setal glands, tenant hairs, molting glands, stink gland, asmataria, androconia, pheromone glands, wax gland, stink gland, lac gland, and milk gland. Now we are going to discuss about endocrine glands, the glands without duct, that is neurosecretory cells, corpora cardiaca, corpora allata, prothoracic gland, and Weissmann's ring. Endocrine glands, these are the ductless glands. They release their hormone directly into the hemolymph, that is the insect blood. The first endocrine glands, the gland without duct, is neurosecretory cells. See here neurosecretory cells in the brain region. Neurosecretory cells are specialized nerve cells present in the brain. They occur as median neurosecretory cells and lateral neurosecretory cell in the middle region, as well as laterally they are there. Their axon joined together form the nervi corpora cardiacae. Nervi corpora cardiacae. See the function of neurosecretory cells, they produce neurohormones, that is PH, TH hormone, prothoracicotropic hormone. Control endocrine glands, other endocrine glands and hormonal activities in insect. Neurosecretory cells secretion controls other endocrine glands and also control hormonal activities in insect. They regulate growth, molting, metamorphosis and reproduction in insects. Next endocrine gland, the gland without a duct is corpora cardiaca. Corpora cardiaca. See, this is the corpora cardiaca. Corpora cardiaca are paired glandular body located behind the insect brain near the aorta. Located behind the insect brain near the aorta, that is part of dorsal blood vessel. They are connected to the brain by means of nervi corpora cardiacae. See the function of corpora cardiaca. This corpora cardiaca store and release neurotransmitter produced by the neurosecretory cells. Help in regulating the heartbeat, maintain tree halos, that is blood sugar level in hemolymph, and participate in hormonal control of the body activities. See next endocrine gland, that is the corpora allata. See, this is the corpora allata. Corpora allata are paired endocrine glands located near and attached to the corpora cardiaca. They are present very nearer to the corpora cardiacum and attached with corpora cardiacum. And this corpora allata secrete juvenile hormone. See the function of juvenile hormone. Juvenile hormone maintains larval characteristics. Larva will be in the larval stage. Prevents premature metamorphosis. They allows metamorphosis after maturity of the larvae only. Regulate molting and development, and important for reproduction and development of accessory gland in adult insects. See the importance. Higher the juvenile hormone level, larvae remain in larval stage. Lower the juvenile hormone level, metamorphosis will be occur in larvae. See here, very important terms related with this juvenile hormone, that is juvenile hormone analog and precautions. Juvenile hormone analogs are the chemicals similar to the juvenile hormone that disrupt normal insect development. See here in clarity, juvenile hormone analog mimic juvenile hormone and falsely maintain the immature stage. In that way, disturbing normal metamorphosis and development in insect. See the effect of juvenile hormone analog. Larva fail to become normal pupa or adult. Abnormal molting will be there. Deformed adult formation will occur and reproduction may be affected. Next, precaution. It is the anti-juvenile hormone chemical that causing premature metamorphosis. Juvenile hormone analogs, they are not allowing for metamorphosis, keeping the larvae in larval stage, but precautions promoting premature metamorphosis. See here, precautions blocks juvenile hormone production so that insects change into the next stage too early, causing premature metamorphosis. Next, prothoracic gland. So this is the prothoracic gland. Prothoracic glands are the endocrine glands present in the thorax region of larval insect. These glands usually degenerated in adult. They secrete ecdysone, also called as the molting hormone. Prothoracic gland secretes molting hormone. See the function of molting hormone that initiate molting in insect, control metamorphosis and growth in insect. See here, what do you mean by precursor? Precursor is a substance that is converted into the another active substance. See example in insect, ecdysone is the precursor. It is converted later into the ecdysterone. Ecdysterone, that is the active molting hormone. Ecdysterone 20-hydroxyecdysone is the active form of ecdysone hormone in insect. See here series. Brain secrete TH hormone, prothoracic hormone that activate prothoracic gland. Prothoracic gland secrete ecdysone, that is a precursor. This is converted into ecdysterone, that is active form of molting hormone that cause molting and metamorphosis in insects. Weissman ring. This ring is formed by the fusion of Corpora cardiaca, Corpora allata, prothoracic gland, and hypocerebral ganglia. Weissman ring is formed by the fusion of Corpora cardiaca. See here, this is the Corpora cardiaca, Corpora allata, prothoracic gland, hypocerebral ganglia. So this is the Weissman ring. To secrete puparium hardening hormone. And this Weissman ring is there in maggot of dipteran flies, that is house flies. Thank you. Thank you all.